Thank you. Good afternoon. As introduced, I'm going to talk about blocks instead of puzzle pieces. And to start, as we all know and experienced as in this workshop, again, science is really collaborative. And we are building on each other's work and really standing on each other's shoulders. And you see here the partners which are just across Europe, which are just involved in this project, most of them actually being here. And in these collaborative efforts, the question always arises, how can we bring our data, our methods, our results together in a more effective way? And to start with the topic of data, and in particular slow waves, with which we are now quite familiar with. Slow waves are interesting here in the aspect that they are really ubiquitous and can be observed, are really observed in across species, across scales, and across methods and simulations, actually representing the 3S in the BESIS acronym. And you see here a few examples on different scales of different temporal and spatial scales. And having these different approaches and viewpoints onto the same phenomenon of slow waves really gives us a great opportunity to basically incorporate them into a cumulative understanding of the phenomenon. However, this also poses some challenge because it's difficult to integrate these heterogeneous sources of data, although bringing them together yields a great benefit in multiple regards. For example, for the integration of the multiple data sources in a meta studies of larger analyses, as well as quantifying the experimental variability that we see, and also for calibrating and validating better models. So in the following, I am going to show you how analysis workflows can support this comparability across domains. And I'm going to do this in an analogy of building space stations, or rather models of space stations. So with analysis workflows as well as space stations, there are basically two ways how to approach this. You could either build them from puzzle pieces or from building blocks. And I'm going to argue that one is better than the other. And as the title already indicates, building blocks are better because building blocks are modular. And they are combinable in multiple various ways, which is not possible with puzzle pieces where only one fits to exactly one neighbor. And this also means that building blocks are adaptable, where you can add, remove, and change individual elements. And you might have experienced this yourself when you collect a puzzle piece, or also an analysis workflow from your cupboard after a couple of years. Then one or two pieces might be missing, and that basically breaks the whole thing. Whereas when you build your space station from building blocks, it's much easier to individually maintain each element and reproduce the whole result. And this maintainability of the individual pieces also means that they are reusable in different contexts, and which also leads to an expandability of the users, so that there's a versatility in expanding the space station across beyond its initial scope. And we're going to see in the following how this applies in practice to workflows. So we implemented this in a pipeline framework for slow waves in particular, called CobraWAP, standing for Collaborative Brainwave Analysis Pipeline. And into this pipeline, the main novelty of this pipeline is that it can integrate multiple data sources, and a few modalities are listed here. And these modalities, data sources, basically enter the pipeline by being represented in a common way. So we are using neo objects to have a common representation, so that all the following steps then can basically be agnostic about where the data comes from. So the building blocks of this pipeline are basically individual methods, which are represented by Python scripts usually with a with a well-defined input-output relationship. And these are also grouped in these sequential stages, thematically grouped, starting from quite general aspects of an analysis pipeline, like processing, and going towards more specific tasks relating to, here, for example, the detection of the up transitions in slow waves. And as you also see here, indicated by these colored points, you can select individual blocks in this pipeline fitting to a specific data set that you input, so that you can adapt the pipeline to the needs of a data set, although the individual blocks are quite general, so they can be combined. So that in the end, when adapting this to various data sources, by going through the pipeline, the heterogeneous data is converged into a more abstract description level of slow waves here, so that in the end we can derive various measures on these various data sets. For example, here we have velocity, direction, interwave intervals. Also, we can use very alternative implementations and definitions of, for example, velocity, basically planar description or local description. And from the viewpoint of the data, I can show you how this looks like. Maybe first to mention, in terms of building blocks, this is also based on a foundation of other open source projects which take care of various aspects of this within this pipeline. Just to mention, for example, Neo and Elephant, which you also see more in detail in the hands-on session later on. So how this looks for the data. So this is inserted as raw data. You see here an ECOG dataset and a calcium imaging dataset provided by EDBubs and Lens. And so these data sets are quite different in terms of spatial resolution and temporal resolution. However, as they are processed, we detect the up transitions of the slow waves locally. You might see this here as dark dots propagating, which are then, while going through the pipeline, grouped together into spatially expanded wavefronts which propagate over the cortex. And this abstract description of groups of transition times is further augmented by here a vector field representing the optical flow of the movement of the waves. And this description level is equivalent between these data sets, and therefore we can then derive a common characterization here, exampled by the dominant wave mode that we see in these data sets. And what this makes this possible is that we can now feed a lot of data sets through the pipeline quasi-automatically and create and basically create meta studies across many data sets and get this overview. So here we have basically 5 data sets with over 6 hours of total activity. And we're still analyzing this, but we can already see some quite interesting results. So here we pooled all this data together and grouped this with respect to their measurement technique and their anesthetic that is used for the anesthetized mice. And something which is, for example, quite obvious for the planarity, so measure how straight the wavefronts are which propagate over the cortex. We see that due to the higher spatial resolution of the calcium imaging, we see that we are able there to detect more complex wave patterns, and therefore just by the spatial resolution, these are less planar than for the ECOG data. And this is more obvious also for the when you look at data sets using isoflurane, because isoflurane is also known to induce waves which are a bit more chaotic, which becomes very apparent here. Further, with calcium imaging, we are also able to detect more waves because of the resolution, also influencing these characteristic measures. that we see a slightly shorter interval between waves because we just can detect more. Further, we see a quite good comparability between for the velocity between different measurement techniques, at least for Cattleman data sets, where a big discrepancy likely also attributed to the difference in resolution and definition of the velocity measure. And further, we can compare direction, where we consistently see that there is a preferred axis along which the waves propagate, although this direction can differ between various setups and subjects. And similarly, first this, and just as a sanity check, we see that we can, the reproducibility that is built into this pipeline allows us to quite feasibly reproduce already existing published results, where we see that the velocity and the interwave interval depends on the deepness of anesthesia, where the deeper the anesthesia level is, the slower the waves become, and also the less frequency waves become. So this motivates basically the reasonable, that the results are reasonable. And similarly, as we can basically use the same methods to analyze different data, we can also use different data analyzed by the same data analyzed by different methods, and therefore see how the choice of methods influences the outcome and the characterization of the wave. Here exampled by two different techniques to detect the up transitions of the slow oscillations, leading to quite different pictures of the resulted wave. And a further application that we see is that since we can also input simulation data into this pipeline, we can apply this for model calibration and validation. And not going into detail here because this will be explained in more detail in a talk tomorrow. But to give an outlook, this further connects to a different project of ours, looking at network level validation, where we basically implemented a library for validation tests on a network description level. So the typical validation test basically compares a simulation against a system of interest, but the same test can also be applied, not basically comparing a model against an experiment, but we can also compare with the same test model against model, or for what we did here, basically using the same model and just changing the implementation, and can therefore validate, so to say, the implementation itself, seeing that numerics and implementation details actually matter for these comparisons. Coming back to the pipeline, the versatility of the pipeline we can further demonstrate by not only applying this to slow waves, but expanding the scope of the pipeline to also apply this to a different kind of wave. So instead of having this propagating up states, we can look at these more oscillatory activity in the beta regime, which also propagates. And we see this, visualize this here as the output of the pipeline based on data from the Rolf Semer lab, quite recently published, and also curated with help from people of Jülich, where you can nicely see various wave patterns across the visual cortex here. This is of a awake behaving monkey, which demonstrates that we can not only make the data of various sources of slow waves comparable, but also we can transfer our methods and terminology to adjacent research community, as for example for beta waves. So to wrap up, I showed you how modularity can be implemented into analysis workflows, and how this can be leveraged to make various data sources comparable and make reproducibility more feasible and easily accessible, while basically laying the groundwork to make to have various additional applications like method benchmarking and reusing of elements to build calibration and validation workflows, as well as extending the scope to different kinds of wave analysis pipelines. So as a takeaway, I hope I could demonstrate that the reusing and sharing of analysis pipelines makes science more collaborative, and that puzzle pieces are, building blocks are better suited than that than puzzle pieces. Thank you.